Hello students, welcome to the last class of genetics. In this class, we are going to study mutation. What is mutation? Mutation is any spontaneous or random changes in the genetic material of a cell or an organism. Mutation can be large scale or gross mutation and point or gene mutation. Large scale or gross mutation is chromosomal mutation that involve changes on the whole chromosome or parts of chromosome due to chromosomal non-disjunction, translocation, inversion, duplication, deletion, and insertion. Large scale or gross mutation are always known to cause serious disorders and hence death. The second type of mutation is point or gene mutation. This is a type of mutation that involves change or alteration of one base pair or one genetic code of a gene. This point mutation can be base pair substitution mutation and frame shift mutation. Base pair substitution mutation is a condition in which one base is replaced by another base that results in silent mutation, nonsense substitution mutation, or missense substitution mutation. The second type of point mutation is frame shift mutation. Frame shift mutation is a condition which is caused by insertion or deletion of bases on a gene. First, let us study point or gene mutation. Point or gene mutation is a condition that only affects a gene. Point mutation involves substitution mutation and frame shift mutation. What is substitution mutation? Substitution is a situation in which one base is replaced by another different base. This substitution mutation can be silent substitution mutation, missense substitution mutation, and nonsense substitution mutation. As we can see from the diagram, silent substitution mutation is a situation in which the base is substituted or changed but has no effect on amino acid sequences of a protein. As we see from the diagram, C is replaced by T. GGC code for glycine and the mutated form GGT also still code for glycine. GGC and GGT are both alternative codes for glycine. Since genetic code is degenerative, glycine has more than two alternative codes. Due to this reason, the mutation has no impact or effect on the amino acid sequences of a protein. Missense substitution mutation is the second way of substitution mutation in which replacement of a base or single nucleotide results in incorrect amino acid in amino acid sequences of a protein. This results in malfunctioning protein or completely different protein. As we see from the diagram, A is replaced by C in the nucleotide sequences. All the codes code for histidine in the nucleotide sequences, but when A is replaced by C, this codes for proline, and therefore the amino acid sequences of a protein are changed and results in completely different protein. Nonsense substitution mutation is the third form of substitution mutation in which replacement or substitution of a base or single nucleotide results in the stop code that cause shortening of protein or cause immature protein formation. As we see from the diagram, when C is replaced by T, the code CAG is changed to the code TAG, which is stop code, and terminate protein synthesis, and therefore this produce immature protein or short protein. I think you have gotten some ideas about the difference between silent, missense, and nonsense substitution mutation. Substitution is not that much serious as compared with frameshift mutation that involves deletion and addition of bases. This is because substitution affects just one triplet, and since the DNA code is degenerative, that means the same protein may still be produced or synthesized. The second type of point mutation is frameshift mutation. Frameshift mutation is a condition when one fewer or one extra base alter the whole sequence after the point mutation, or where a base or a genetic code is added or deleted. Frameshift mutation can happen due to deletion or addition of base or a triplet code. In this case, frameshift mutation can be deletion mutation and addition mutation. Deletion mutation is a situation when a base is missed out during replication, and sometimes a whole triplet or one extra or fewer code is missed out. Whilst in addition mutation, an extra base is added or a whole triplet is inserted. Both these are more significant mutation than substitution. In this case, totally different mRNA is produced that results in a non-functional protein or no protein at all. Example, let us see a sentence that can show us frameshift mutation with a reading frame of 3 letters like triplet codes of a gene. It becomes the man was hot and ran for his hat. As we can see, each word of a sentence contains 3 letters. But if we take out S at the end of was, that is a deletion mutation, it becomes the man, wah, ota, ndr, anf, orh, ish, and at. In other words, it no longer makes sense. In genetic term, it is missense coding. As we can see from the diagram, the original DNA codes for an amino acid sequences are CAT, 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 and CAT that all codes to histidine. If A is deleted at the fourth triplet code, the reading frame is completely changed and results in completely different and malfunctioning protein. Let us see from the diagram how the reading frame gets changed due to deletion of base A and produce completely different amino acid sequence after where base A is deleted. CAT, CAT, CAT. When A is omitted, the reading frame can be CTC, that codes to leucine, ATC, which codes to isoleucine, ATC codes to isoleucine, ATC again codes to isoleucine. This shift of reading frame results in incorrect amino acid sequence, which may produce a malfunctioning protein. Insertion mutation is other forms of frameshift mutation in which a base or a triplet code is added or inserted into the original triplet code or base sequence and results in shifting of triplet code reading frame. Therefore, it produces incorrect amino acid sequence which may produce a malfunctioning protein. As we see from the diagram, the original DNA code sequences are CAT, 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 and CAT, which codes to a protein which has histidine amino acid sequences. But when A is inserted into the fourth DNA code, the reading frame is automatically changed and produces incorrect amino acid sequence. The changed DNA code is CAT, 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 ACA, TCA, TCA, and TCA. CAT code for histidine, CAT code for histidine, CAT code for histidine, ACA code for threonine, TCA code for serine, TCA code for serine, and TCA code for serine. These amino acid sequences are significantly different from the original amino acid sequences in a protein. So, insertion of a single nucleotide results in incorrect amino acid sequence, which may produce a malfunctioning protein. Causes of point mutation. The rate of mutation can be increased by a number of factors or mutagens, including spontaneous mutations. These are mistakes that accidentally occur when DNA is replicating. The second groups of mutagens or factors can be carcinogenic chemicals. These carcinogenic chemicals are chemicals that can be found within tobacco smoke, reactive oxygen molecules with free radicals, benzene, and certain viruses. The third factor is high energy radiation. This high energy radiation includes ultraviolet radiation, X-rays, gamma rays, and alpha particles that are capable to change the chemical natures of DNA. We have to remember that, which is quite surprising when you consider that each cell contains 6 billion base pairs that might mutate. Biologists estimate that mutations arise at the rate of 1 in 50 million base pairs. This means that each new cell will have on average 120 mutations. This doesn't worry us, but we should remember two things. Most of these mistakes or mutations are detected and repaired by proofreading enzymes. And since 95% of our DNA is non-coding, most mutations are unlikely to affect the coding genes. That means most mutations are more likely to occur on this 95% of our non-coding DNA. These are the reason that most people are less vulnerable to genetic disease. What are the consequences of gene mutation? The consequences of gene mutation depends on the type of cells and the genes where this mutation occur. If this mutation happen on active or more fundamental cells and genes, the problem might be serious. Mutations on normal body cell will have one of the four possible consequences. The first consequence can be it will be completely harmless, will be damaging or kill the cell, it will make the cell cancerous, or it might kill the whole organism. Somatic cell mutation might affect no other person since it cannot be passed on the next generation. But sex cell mutation might be passed on to the next generation and affect the whole family tree. Mutations in different genes have different effects, but two types of genes are really important. These are proto-oncogenes and tumor suppressor genes. Proto-oncogenes always regulate cell division, whereas tumor suppressor genes prevent the formation of tumor. When proto-oncogenes mutate, it often become active oncogenes. These oncogenes stimulate infinite cell division or it initiates the cell division to become uncontrolled. Normally or ordinarily, cell division stimulated by external growth factors. We know that tumor suppressor genes recognize uncontrolled cell division and act to suppress this uncontrolled cell division. If this tumor suppressor genes gets mutated, it becomes inactive. In this case, the tumor will form due to uncontrolled cell division. Do you know what does tumor means? Tumor is a mass of cells created when cell replication gets out of control or cell replication becomes infinite. Tumors known to cause a disease cancer. What are the importance of mutation? So far, we have talked only about how mutations produce harmful effects, but sometimes mutation can produce certain values. The values are since mutations are the raw materials of evolution that can let organisms evolve or get changed over time. Because mutation is the only process that can create new genes, which is important for the evolvement of organisms on Earth. But the other processes like crossing over, segregation, random assortment, and random fusion simply reshuffle the existing genetic material. If a mutated allele gives an advantage to an organism, then natural selection acts on it and let the frequency of that allele increase with successive generations. In this case, numbers of organisms that have that mutated genes also increase. For example, mutations in the DNA of bacteria can give them resistance to specific antibiotics such as penicillin and ampicillin. In 1947, the first penicillin-resistant bacterium was found. This bacterium was called Staphylococcus aureus. Today, over half the infections caused by Staphylococcus aureus are caused by penicillin-resistant types or strains. Really surprising that bacteria can swap or exchange antibiotic resistance genes with each other. Most of the mutant genes that confer resistance are found in plasmids. They can transfer these plasmids to other bacteria by conjugation, transduction, and transformation. Through conjugation, the plasmid passes through a special conjugation tube from one bacteria to the other. In case of transduction process, a virus carries the plasmid from one bacterium to another. Through transformation process, the plasmid is absorbed from a dead bacterium. Chromosomal or gross mutation. Chromosomal or gross mutation occurs when there is any change in the arrangement or structure of chromosomes. Chromosomal mutations occur most often during meiosis at crossing over in prophase 1. Chromosomal mutation are much bigger events than point mutation and usually results in the death of a cell. This may also affect the whole organism. For example, if essential parts of the DNA are affected by chromosomal mutations, a fetus may be aborted. Chromosomal mutation can occur due to inversion, deletion, insertion, duplication, chromosomal non-disjunction, and translocation. Chromosomal mutation due to inversion occurs when an area of DNA on a chromosome reverses its orientation on the chromosome. As we can see from the diagram, we can suppose a chromosome that has gene sequence A, B, C, D, E, F, G, H, I, J gets its part that contain gene C, D, E, F inverted by 180 degree. The resulting mutated chromosome could be a chromosome that has A, B, F, E, D, C, G, H, I, J. One inversion on chromosome 16 can cause leukemia. An inversion that leads to an embryo having too few or too many copies of genes can cause the embryo to miscarry or fail to grow or born with substantial medical problems. With this cause of mutation, a decrease in the number of genes occurs due to deletion of large section of a chromosome. Deletion can result in a variety of genetic disorders such as Prader-Willi syndrome, results from deletion of parts of chromosome 15. Prader-Willi syndrome happens due to malfunction of the hypothalamus. Hypothalamus is a small endocrine organ at the base of the brain, which plays a crucial role in many bodily functions, which means hypothalamus is responsible to control hunger and satiety, temperature and pain, fluid balance, puberty, emotion, and fertility. Insertion type of mutation describes an increase the number of genes caused by when unequal crossover happens during meiosis. The chromosome may become abnormally long or short and stop functioning as a result. As we can see from the diagram, part of chromosome 4 cut out and inserted to chromosome 20. In this case, chromosome 4 becomes abnormally short and chromosome 20 becomes abnormally long due to insertion mutation. Duplication as cause of chromosomal mutation results in the genes displayed twice on a single chromosome. These are usually harmless as the chromosome still has all its own genes. However, duplication of the whole chromosome is more serious. For example, having 3 copies of chromosome 16, what we call trisomy 16, leads to babies being born with a range of medical issues such as poor fetal growth, muscular and skeletal anomalies, congenital heart defects, and underdeveloped lungs. As we can see from the diagram, genes ABC on the chromosome duplicate and displayed twice on the chromosome as ABC ABC. Chromosomal non-disjunction is another situation when homologous chromosomes do not separate or segregate successfully to opposite poles during meiosis. That means one of the gametes lack a chromosome and the other gamete have an extra chromosome. If this chromosomal non-disjunction happen on chromosome 21, it results Down syndrome. A person who has Down syndrome contains 47 chromosomes in each cell. That means each cell contains 3 copies of chromosome 21. Down syndrome is characterized by mental retardation, heart defects, and stunted growth. In translocation chromosomal mutation, a piece of one chromosome is transferred to another non-homologous chromosome. These types of chromosomal mutation are responsible for the occurrence of chronic myelogenous leukemia. Chronic myelogenous leukemia due to translocation mostly occur on Philadelphia chromosome. That means this can happen when genes translocate from chromosome 22 to 9.